Hello everyone, my name is Shalini. I'm a second year dental student at Roseman College of Dental Medicine. I am working on publishing my undergraduate thesis in the field of astrobiology and developmental biology. My project was on oogenesis in tardigrades. So out of these pictures, which one do you think represents a tardigrade? Obvious answer is all of them. There are about 1,200 species of tardigrades, each different from the other, and they're about 200 micrometers in length. They're commonly known as water bears, and they're extreme tolerant, meaning they're able to withstand extreme temperature, not necessarily love them like extremophiles, but can withstand them. They're found all along the globe, in the Arctic, in the rainforest, and anywhere in between. And they do this by going into a tent state and coming back from the dead. So the tent state is when it's cryptobiosis, when you desiccate them or when you dry them, when you put them in low temperatures, when you put them in high osmotic pressures, they go into this inactive state, and then when conditions are normal again, they come back to life, which makes them very interesting. And the species that I worked on is Hypsibius dujardini. I know it's a bit of a mouthful. They're transparent, they're freshwater dwellers, but they're low desiccation. They have a low desiccation tolerance, meaning they don't do so well as other tardigrade species with desiccation, and they're parthenogenic. Parthenogenic stands for if there's no males present, these females are able to reproduce on their own, which is great in a model biological organism. So this is the reproductive anatomy of Hypsipias, and you can see the gut over here, the ovaries, and it's really helpful that these species are transparent and exactly why they were chosen for the study. So this female is about to lay eggs, and she usually sheds her exuvium, just like a snake sheds her skin. And the eggs are laid inside the skin for protection. So eggs hatch about 4 to 5 days after being laid, and those hatchlings reproduce 6 to 7 days after, and then again 2 days after. So the gestation period after the first one is very short. And oogenesis, what does that mean? So oogenesis stands for the formation or the beginning of eggs, so the development of eggs. And that has been previously studied in other species, but not in Hypsibius, which is exactly why I wanted to do this project. And I wanted to take it a little bit further by doing a cold tolerance test and see how that affects oogenesis. So my study was divided into 2 phases. The first one was pretty simple. It was just to observe how oviposition events or egg-laying events occurred at room temperature. And then the second was to see them at a colder temperature, see if there was any difference. So the first phase, 25 individuals were selected on the basis that there were no eggs inside the mothers that were visible. And then I observed them for 20 more days after that, every single day. And the results showed that oviposition occurred in intervals, some days being more popular than others. For example, day 4 and day 6, there were a lot more eggs than day 5 and day 8 among all the females. This maybe indicates that there's some sort of synchronous egg-laying event happening within a population. And the differences in clut size did not impact when the next oviposition event was going to occur. And every single individual that was selected did lay eggs, but some of them laid them up to 4 times. So there was big differences between individuals. So the second phase, I chose 8 individuals from the first phase of experiments, and then I lowered the temperature 10 degrees at a time, and eventually observed them for 9 days in total to see if there is any difference in egg-laying behavior. So this graph shows that the 8 individuals that were selected laid eggs during normal temperature, in normal temperature. And then in colder temperature, only 2 of them ended up laying eggs, that too at the beginning of the experiment. So one can speculate that fewer number of eggs produced could be because the metabolism is slower, all the energy is put into survival rather than reproduction, which makes sense. And the eggs that were laid in the beginning of the experiment could just be that they were formed when the females were still in the room temperature. And no further egg formation occurred. So that's a pretty interesting find. And what does all of this mean? Why do we even care about this? like I said, they're great model organisms because they're extreme tolerant. And by understanding how they're able to stabilize their biological molecules can lead way, for example, we could use it to stabilize vaccines. We could make stress tolerant crops if we know how that works better. And they're also able to withstand radiation in space. So this is a great way to learn what the limit for life is and if life can exist outside of Earth. And when I was doing this project, I found that there was a lot of gap in the development of biology, especially for Hypsibius. So my hope was that this project is just a small piece in a bigger puzzle. And in the future, hopefully it leads way for more studies on this topic. And I want to thank Dr. Stone for supervising this project from McMaster University, and Dr. Hung from Roseman University for potentially helping me publish this paper, and for the INSTAR committee for giving me this opportunity to present. And I hope you have a great rest of the symposium. Thank you.